In this video, we will talk about the structure of DNA. As you know, the DNA or deoxyribonucleic acid is the genetic material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. It is the hereditary material as it bears the genes that are passed from parents to offspring, and it provides instructions for growth and functioning of living organisms. DNA is known as deoxyribonucleic acid because the pentose sugar in DNA is deoxyribose. It means that the 2' carbon in this sugar lacks a hydroxyl group, which is replaced by hydrogen. As in this figure, you can see that the 2' carbon contains a hydrogen group. RNA possesses ribose sugar as it contains a hydroxyl group at the 2' carbon of the ribose sugar. Next, DNA is a large molecule and it is made up of subunits called nucleotides. A nucleotide basically consists of a nitrogen containing organic base that can either be a double ring or single ring. So here in this figure you can see that this is a double ringed nitrogenous base, whereas this is a single ringed nitrogenous base. The double ringed base is basically purine that includes adenine and guanine, whereas single ringed base is pyrimidine that includes guanine and cytosine. So the base is linked with the 1' carbon of the deoxyribose sugar. Here in this figure you can see that this is 1' carbon of the deoxyribose sugar, and at the 1' carbon the nitrogenous base is attached via the nitrogen beta glycosidic linkage. So nucleotide also contains a pentose sugar that has already been discussed, pentose sugar that is deoxyribose, and a phosphate group. This is a phosphate group, and the phosphate group is basically attached to the 5' carbon of the deoxyribose sugar via the phosphate bond. So the 1, 2 or 3 phosphate groups can be attached to the 3' or 5' carbon of the deoxyribose sugar. So now what is the difference between a nucleoside and a nucleotide? Nucleosides contain only sugar and the nitrogenous base. Okay, so whereas in case of nucleotides, the nucleotides contain the sugar base and a phosphate group as well. So next is the condensation reaction. The condensation reaction is basically a reaction that occurs between nucleotides and results in the formation of long chains. DNA is a ladder-like structure, and the rails of the ladder basically consists of alternating sugar phosphate groups. So in this figure you can see that the phosphate group of a nucleotide attaches at the 5' carbon of the sugar. So here in this figure you can see the phosphate group is basically attached at the 5' carbon of the deoxyribose sugar. So the adjacent nucleotides attach to one another by covalent bond between the 5' carbon phosphate at one nucleotide and the 3' carbon of the deoxyribose. So this is a reaction that occurs between the 3' the hydroxyl group that is attached to the 3' carbon of the deoxyribose and the phosphate group that is attached to the 5' carbon of the deoxyribose. So in this way the nucleotides, adjacent nucleotides, are linked to each other, and this reaction is known as condensation reaction, and it results in the formation of phosphodiester linkage, and a water molecule is released as a byproduct. So next is the pairing of nitrogenous bases between the two strands that hold them together. So the two strands of DNA are held together by the pairing of nitrogenous bases. So in this figure you can see that adenine is hydrogen bonded to its complement thymine by the formation of two hydrogen bonds. So Guanine is also hydrogen bonded to its complement, that is cytosine, by the formation of three hydrogen bonds. So these two DNA strands are oriented in opposite directions so that the 3' carbon of the deoxyribose in one strand, for example here, the 3' carbon of the deoxyribose in one strand is oriented opposite to the 3' carbon of the other strand. So here, so the orientation of these 3' carbon in the strand is opposite, and the two strands have opposite polarity, and DNA molecule is said to be anti-parallel. So the entire molecule is twisted as a right-handed helix, and one complete spiral contains 10 base pairs. So this gave rise to major and minor grooves. Here in this figure you can see this is the major groove, this is the minor grooves in the DNA strands, and a single helical turn of the DNA stretches over a distance of 34 angstroms, whereas the distance between the two base pairs in the DNA is 3.4 angstroms. So this was all about the structure of DNA.